Hello everyone, in this module we'll be discussing about the biology-based data. Most of the biology-based data available is in the form of DNA, RNA, proteins and metabolic pathways. We know that the cells are the building blocks of life. A cell is a chemical system which is able to maintain its structure and reproduce on its own. Coming to the differences between the prokaryotes and the eukaryotes, the prokaryotes being the primitive organisms have rigid cell walls and the DNA is found in cytoplasm in a region called as nucleoid. Whereas in eukaryotic cells, the cells are separated by a cell wall in plants and by a plasma membrane in animals. They have a well-defined nucleus with DNA wrapped around the histone proteins. This is the overview of the eukaryotic and the prokaryotic cells. The structure of DNA is discovered by Watson and Crick in 1953. It is a double helical structure with complementary base binding. It has four bases: adenine, guanine, cytosine, and thymine, which form the building blocks of DNA. Adenine binds to thymine with a double hydrogen bond, and cytosine binds to guanine with a triple hydrogen bond. Both the strands of DNA are antiparallel to each other. This is a basic structure of DNA with phosphodiester bonds forming the backbone and sugar molecule bound to the bases. So what exactly is a gene? Gene is a part of DNA that can translate into a protein. During transcription, thymine in DNA is replaced by uracil in RNA. The three RNAs, mRNA, tRNA, and rRNA, play a very important role in translation. Every three bases in mRNA form a codon and codes for a specific amino acid. Every three bases in tRNA has specific anticodon that binds to a specific codon. rRNA is a component of ribosome and helps in translation. Here is an example of prokaryotic and eukaryotic gene. We can see that the genes in prokaryotes are a continuous stretch of codons, whereas in eukaryotes, a gene has two regions, the red-colored coding region exon and the white non-coding region intron. After the formation of RNA, the non-coding introns are spliced out of the RNA. These are the main differences between the prokaryotic and the eukaryotic gene expression. Detecting the coding region of a sequence is called as gene prediction. It is very simple in prokaryotes as a gene can be identified using the open reading frames, whereas in eukaryotes, a gene is a mix of introns and exons. Therefore, a gene can be identified by hidden Markov models for gene finding. All these tools are available online, and I have provided the links for GenScan and Glimmer. Translation completes the flow of genetic information within the cell. The sequence of nucleotides in DNA has now been converted into a sequence of amino acids in polypeptide chain. The synthesis of polypeptide chain, however, is not equivalent for the production of a functional protein. To be useful, the polypeptide must fold into distinct three-dimensional conformations by interacting with the side chains of their constituent amino acids. The structure of protein is usually determined by X-ray crystallography and NMR spectroscopy. Foldit is an online game on protein folding which will help us further to understand better about the protein folding. Now that we know how the information in DNA is passed on to mRNA and proteins, we can fetch the information of various cellular activities by using the science of biotechnology. DNA hybridization is a process in which the DNA of two organisms are incubated to allow the DNA strands to unwind and rebind, forming a hybrid DNA. The hybrid DNA with higher degree of similarity binds more firmly, and thus DNA hybridization is used to determine the evolutionary distance of two species and is used in making the phylogenetic trees. The second approach is a polymerase chain reaction. PCR can make billion copies of a target sequence of a DNA in very few hours. It follows three simple steps: DNA denaturation, primer annealing, and DNA polymerization. PCR has many applications. For example, in forensic medicine, it is used in a process called as DNA fingerprinting. It is also used in archaeology and paternity testing. Gene expression analysis is gaining importance because any change in the cell's internal or external environment can lead to changes in the gene expression. Most of the human diseases manifest through a misregulation of gene expression. Here is a list of the techniques which are used to detect the gene expression levels. Out of all these, microarrays, SAGE, and cDNA libraries are the high throughput techniques used to estimate the gene expression levels. Protein-protein interactions are very important in many of the biological processes. They help to decipher the molecular mechanisms underlying the biological functions and enhance the approaches for drug discovery. Yeast-2 hybrid method and affinity purification are the two most important methods that are used to determine the protein-protein interactions. DIP and MIPS are the two databases which maintain a complete list of the protein-protein interactions. A metabolic pathway is a series of chemical reactions occurring within a cell catalyzed by an enzyme, resulting in the formation of a metabolic product to be used or stored by a cell or a precursor of another metabolic pathway. Metabolic pathways help to regulate the gene and protein expression and also can mediate the signaling between the organisms. These are the public resources available for the pathways. Reactome is a curated pathway database, whereas KEGG includes the metabolic and regulatory pathways, and Metapsych is a non-redundant database. I would suggest everyone to go through the information and the learning goals in our wiki page to know more about the biology-based data.